Well, good morning, everybody. Oh, thanks very much. Don't applaud until after you've sat through 45 minutes of this. So, okay, as the lead-off speaker for the scientific session today, my job is to introduce you to a semester's worth of neuroscience in about an hour, and to give you sort of the language and the terminology, the set of ideas which are then going to underpin the other talks that come. So you have a sense of what it means when an axon connects to a synapse and connects on to an next neuron, the basis of some of the neuroimaging technology we're going to talk about. So I will say I'm just going to get used to the challenges of this projection system. But let me figure out where I am and how to advance. Oh, that looks like that's going to work. Okay, great. So we're on the first slide. All right, perfect. So I imagine that many of you, as did I, flew to attend this conference today. And as is my habit, since I have to turn off my iPad during the takeoff, I often look through the SkyMall catalog right on the takeoff. So the SkyMall catalog, I have recently discovered, now caters to aging neuroscientists, because if you look in, they now sell these magnifying glasses, whole-page magnifying glasses. And if you zoom in on what this retiree in Florida is reading, it's actually a neuroimaging study. So that's what the SkyMall catalog is selling now, which really says something about neuroimaging perhaps has reached its peak. It might be a warning sign for your field if it's now featured in SkyMall catalog. But it really does speak to how ubiquitous these neuroimaging studies have become in common discourse. You see them all the time in see them frequently in Time and Newsweek magazine. The reporting about the results of different neuroimaging work really started to pervade into all corners of society. And as this conference attests, law is no stranger to these topics as well. And from the SkyMall catalog to something perhaps a little bit more relevant to this group, what's shown here are the images from a report which in this case appeared in Nature magazine, but was describing the introduction of neuroimaging evidence in a criminal case. And this was at the sentencing phase for this character here, Brian Dugan. So I can see what I'm doing too. Okay. It was the sentencing case for this character, Brian Dugan, who had been accused of killing an Illinois police officer. I'm sure to many in this audience this is a familiar story. In any event, a neuroscientist, Kent Keel, testified at the sentencing phase that there were neuroimaging markers, so there were patterns of brain activity seen in this defendant, which indicated that he was a psychopath, and that this was a feature of his makeup. Now, leaving aside for a moment whether or not his identification, the defendant's identification as being a psychopath, should be a mitigating factor or not, it might raise the claim. It might raise the question of how we should take evidence like this, neuroimaging evidence, as suggesting that this individual himself is a psychopath. And that is a sort of a motivating case study that I'll return to as we go through this talk. But to start with, I'd like to sort of move us from the broadest reaches of organization of the cortex down to some finer scale details, and then we'll make our way on over to neuroimaging. And throughout this, what I'm going to do is try and give you sort of a cartoon view of the organization and general principles of how neuroscience works, and then occasionally pull back the curtain to let you see some of the raw horror that is all the tiny details that make up the actual implementation of the science. So the first thing is, let's just get an overview of the organization of the brain, right? So which is on the left and what's on the right, and how these different structures are associated with different kinds of behavior. So what you see there on the screen is the view of the left side, or the left hemisphere of the brain seen from the side view. And we're going to take a little tour of the different lobes and organizational components of this structure. So the first thing we can do is take a look at sort of a cross-sectional view down through the brain. This is what's referred to as a coronal slice, and we'll come back to these different slices and views that you can have for the brain in a moment. And on that coronal slice, you can see that there are different structures which are readily apparent just at the sort of the grossest level, just by looking at it. And the brain generally is divided into gray matter and white matter, and into gyri and sulci, or individual gyruses and sulcus. So the gyrus is the fold that the brain is cast into, sort of wrinkled up like a walnut. So the lumpy bits are the gyrus, and then the sulcus is the depths or the valleys in between those wrinkles. Then the gray matter is the one substance of the brain which surrounds it on the surface, whereas the white matter are the deeper portions in. And generally speaking, that white matter also goes by the name of the cortex. So those are the two big divisions, the white matter and the gray matter. within the cortex lives the metabolically active cell bodies of neurons. So let's take a closer look at a neuron. So a neuron is the microscopic cellular component that makes up the central nervous system in the brain. And an individual neuron has a general structure of including a cell body at one end, which is shown there. Let's see if my pointer works. Can you actually see a pointer on the screen if I move that around? Yes pointer, no pointer? No pointer. Okay. You can see that there's a cell body, which is the rounded end, which then leads to a long tube that casts away. So the cell body at one end, and then what's called the axon is the long connector that goes up away from the cell body. So the nerve cell is the fundamental unit of computation and communication in the brain. The cell body is where all the metabolic and cellular activity takes place, and the axon is the communication cable that connects one neuron to another. So located within sort of the structure of the brain, the cortex, or that gray matter layer on the outside, that's where the cell bodies live. So the cell bodies are all positioned there. And then the axons stretch down through the white matter to connect to other neurons in different parts of the brain. Okay? So the gray matter contains the cell bodies, and the white matter contains the axons. Now, of course, the complicated truth is far worse than what the simple model would suggest. In fact, the cortex is composed of multiple layers, and in those layers there are many different sorts of types of cells, and they're interconnected both within the layers of the cortex, and then the cells come in a variety of different forms with different sorts of structures to them and different kinds of connections. But in general, we can stick to this simple model of each nerve cell has a main computational hub, which is the cell body, and then a long connection which reaches out to connect to the next cell, and that those connections take place in the white matter. Okay, so now we have this idea of having individual nerve cells. How do they communicate to one another? So the core unit of communication from one nerve cell to another is what's called the action potential. So the action potential is an electrical discharge that travels down that long axon cable from one nerve cell to another. So it works a little bit like this. At one end, on the cell body, are what are called the dendrites. Those are those small little projections that come off of the rounded end of the cell body at one end. And that's where the prior neuron has extended its axon and connected to the next neuron. Right? So the dendrites are where you receive input from the prior neuron. And those dendrites, when they receive stimulation from earlier neurons, can sum up that input activity and then produce what's called an action potential. This discharge, this spike of electrical activity that then travels down the neuron along its length. Okay? That reaches all the way down to the very end of the neuron into what's called the synapse. And the synapse is the point of connection from the end of one neuron onto the next one. Okay? And that electrical transmission of activity is rather rapid, as one might expect for a movement of electrical charge. So those neurons are then connected together in large networks of cells, right? So one cell body, one neuron, then extends its axon out to connect to the next neuron, which then extends its axon out to connect to the next neuron, and so on in a series of webs and chains of neurons that make up then the structure of the brain. So what I'd like to do then is focus in on this one particular part of this connection, that what's called the synapse, the point of contact from where one axon reaches down and has the synapse that then connects on to the next one. So if we're going to zoom in on that now, and what we'll see then is the axon coming down to the synapse, which is that little bump at the end. That's the first neuron, and then the second neuron, which is the dendrite of the next neuron right after it. Okay. So when you zoom up in real close, what you find is that at the end of that axon, in that point of the synapse, is a whole bunch of what are called vesicles. They're little bubbles, and inside each little bubble is a collection of chemicals referred to as neurotransmitters. And when the action potential travels down the axon and hits that synapse, that causes those bubbles to move down and line up right at the edge of the synapse, right? To reach out and get up to right at the edge towards that gap between one neuron and the next. And then those bubbles break open and release that chemical onto the next neuron, okay? So this chain of events then is that an electrical potential travels down an axon real quick. That's an electrical transmission, so fast down the axon, hits the synapse, all the little bubbles moved right towards the edge of the synapse, release the chemical, which then has to travel across a small gap where it's picked up by and grabbed onto by the next neuron, and thus continuing the chain forward, okay? So rapid electrical activity, synaptic transmission of sprinkling of chemicals onto the next neuron, then that triggers off the next electrical spike to travel on down the chain into the next neuron. Okay? So fast electricity and then slow chemical transmission. So just as I had mentioned, so they transmit across, are grabbed onto by receptors. So in that what's called the postsynaptic neuron, so in the dendrite of the next neuron, you have these individual little receptors. These are like little catcher's mitts that catch the individual chemicals that are coming off of the prior neuron. And then those receptors are matched up for which particular neurotransmitter is coming across. And then that produces the electrical stimulation that then moves on to the next one. Okay? So now that general principle holds throughout the nervous system. You have a neuron, electrical transmission, release of neurotransmitter, and then on to the next one. But of course, the messy detail and what gives rise to the enormous variability in the form of computations that nerve cells can perform and the various behaviors that neurons can have comes from the variety of neurotransmitters that we have, right? So there's not just one neurotransmitter, but there are many, many different types of neurotransmitters, right? So each a different kind of chemical, which has its own particular distribution across the structure of the brain. So some of those neurotransmitters might even be familiar to you, right? Those are names you've heard of before. Things like dopamine, right, which will be mentioned, I think, a few times during this course. Serotonin, acetylcholine, all different chemicals that serve for different types of neurotransmission. And additionally, of course, these different chemicals and their release and their reuptake and how they act on different neurons, these are all the most common targets of medications, right, which both treat different kinds of common disorders of mood and regulation, but a lot of different diseases, right? So there are a number of different neurologic and psychiatric diseases which have as their manifestation an alteration of these neurotransmitters, either their distribution or their quantity, and so that's often a frequent target of drug development and treatment. And it even gets worse than that because there's not just a single type of receptor. There's actually many different types of receptors. Some receptor types are excitatory, and some receptor types are inhibitory. So sometimes a neurotransmitter hits the next neuron, and it causes the neuron to increase its activity. And sometimes it hits the next neuron and causes the neuron to decrease its activity. But again, in general, we can boil all this complexity down to electrical transmission along axons, the release of neurotransmitter, which then leads to some change in the next neuron along the chain. So as I'd mentioned, most of the metabolic, so like the consumption of energy activity that goes on in these nerve cells takes place at the cell body. So you can think of each of these nerve cells as little chemical factories. This is where inside the nerve cell it produces proteins that make up the structure of the cell. It turns out that the recycling and use of these different neurotransmitters requires energy. Just the general metabolic functioning of the cells requires a constant consumption of both oxygen and glucose, so glucose being the primary fuel that nerve cells use. So all that activity takes place right in the cell body. So to a first approximation, the axons themselves really don't consume any kind of oxygen and fuel or glucose, but that the cell bodies do. That's where most of this activity is taking place. Okay? So just to get an interim summary from our basic neuroscience and neurochemistry that we've covered so far, we have that the cortex, which is referred to also as the gray matter covering, right, contains the neuron cell bodies. The white matter contains axons, or the connections, right, between one cell body and the next. And the cell bodies are the location where the nerve cells consume glucose and oxygen, right, to remain constantly metabolically active. Indeed, the nerve cells of the brain are some of the most metabolically active cells in the entire body. So there's a constant demand for oxygen and glucose by these cells, and thus very sensitive to any kind of drop in the amount of oxygen or interruption of blood flow. This is why it would be so easy. The first thing that somebody does when they don't get enough oxygen is to pass out, right, because the function of the nerve cells is interrupted then. And then these neurons communicate both electrically and chemically, right? So electrically very quickly down the length of an axon, and then chemically with the transmission of neurotransmitter from one cell body to the next. Okay, so I think that is what we need to know down on the microscopic level of the function of these nerve cells to get us then going to the next stage, which is to zoom way back out and now take a look at this macroscopic organization of the cortex, how the brain is thrown into different lobes and structures which subserve different functions. Okay, so once again we have our side view. So here we go. This is now a lateral view, side view of the brain looking at the left hemisphere. And what I'd like to do is walk our way through both the take a look at both this lateral or side view of the brain, as well as the more mesial or inner side of the brain. So what we're going to do is take one half of the brain, right? So the brain comes in two lobes, the left and the right side. We're going to break that into the two different hemispheres, and we've rotated around one of the hemispheres so that we can see now on the interior side. So in addition to the big folds that you can see that characterize the gyri and sulci, right, the big folds of the walnut that make up the surface of the brain, there are some substructures in there which have particular names and have been assigned different kinds of functions. So let's walk our way through these. So in the very back of the brain, so located back here just behind the rear of your scalp, is the occipital lobe. And the occipital lobe is the structure in the brain that's most focused for the analysis of vision. So information from the eyes travels back to this portion of the brain, and here resides all of the complicated computational machinery which allows us to make sense of the visual world and interpret what we see. But then coming away from the occipital lobe are a number of other regions which also have specialized functions. So moving now towards the top of the brain in blue is what's called the parietal lobe, so located roughly up here. The parietal lobe has some specialization for attention and spatial perception. So damage to this region of the brain causes the neurologic phenomenon of neglect, being unable to tell where things are in space or pay attention to parts of the world. So the parietal lobes represent where things are located in the environment and also have some function in language that I'll come back to in a moment. So the frontal lobe is this enormous expanse of brain all towards the anterior part. So from about the crown of your head forward is the frontal lobe. And of course, as I'm sure many of you heard, this is a very distinct feature of not just the primate brain, but the human brain per se. So compared to other mammalian species, we have an enormous frontal lobe compared to other animals. Now, this is a part of the brain that is specialized both for language production, as well as a set of very higher level and difficult to describe behaviors which we could refer to as planning or executive function. So this is the portion of the brain that helps inhibit the automatic behaviors you might want to make, that helps you plan and sequence actions. Okay? So the frontal lobe generally serves those functions. And finally, the temporal lobe, which is illustrated here in green, has a number of functions involved in hearing and aspects of vision, but really the thing that's most relevant for this audience is that this is the site of structures which are most intimately involved in the formation of memory. So within the temporal lobe is located a structure called the hippocampus, which you'll hear more about, I'm sure, over the course of these lectures. And the hippocampus is a critical structure for forming and storing long-term memories. Okay? So in addition to these general parcellations of functions into different regions of the brain, what I can provide for you is sort of a series of kind of left, right, back, front, top, down dichotomies, which kind of help you keep straight which function goes where, right? So there's an awful lot of different things that you could assign to one corner of the brain or another, an awful lot of different mental operations that would be hard to memorize, that this bit goes there and that bit goes there. But here's a few different rules of thumb, which will at least get you in the right neighborhood of chunk of brain when you're thinking about a particular mental operation. So let's start with left-right. Okay, so now, first off, I know there's an awful lot of stuff out there about left-right. You've heard that, oh, if the right side of your brain is more developed, you're more creative. Isn't that it? And then the left side is you're more analytical. That actually has very little mapping to reality. Here's the truth about left and right. So left and right, there is some specialization. So in the left hemisphere of the brain is a predominant representation of language. This is true for just about everybody, both right-handed people and left-handed people. There is a minority of left-handed people who have these functions reversed. But for most folks, the left hemisphere of the brain contains representation of language and actually some knowledge of skilled use of the hands, how you know how to use tools and how a hammer works and things like that. The right side of the brain, however, is more specialized for spatial representation and attention, your ability to track multiple things moving through the environment, more of a right hemisphere function. There's another fundamental division and organization of the central nervous system, which is front-back. And it turns out that the back portion of the brain is for perception, and the front half is for action. So in general, nervous system input, right, things that you hear, things that you see, things that you feel, all that eventually arrives towards the back portion of the brain. And then when you seek to then make an action, to move your arms and your legs, right, to plan and act, that all tends to come from towards the front of the brain. So an action-perception divide. There's also a division between the top and the bottom, which relates to storing information about where things are in the environment and then down lower, what things are, right? So at the top half, you might represent where somebody is standing, and the bottom half might represent who that person is. And finally, there is a division, and this was a slide suggested to me by my colleague Oliver Goodenough, who's here in the audience, a division between the outer portions of the brain and the inner, more deeper portions. So generally speaking, the cortex, or the outer portion of the brain, is involved in more deliberate or conscious mental operations, whereas there are a number of structures that are located deeper in the brain, what are called subcortical structures, which are involved in more automatic or unconscious processes. And some of those structures are labeled in red, and you'll be hearing more about those in talks coming up. Things like the nucleus accumbens and the amygdala are structures that are involved in things like desire and emotional states, and those are located more deep in the brain. The cortex on the outer surface are structures that are more generally associated with deliberative and conscious behaviors, okay? So, an interim summary for where we are now. So there is regional specialization for mental function, right? And I think this is a fundamental discovery and refinement that's taken place over the last 200 years of neuroscience, is that the brain is not some big, uniformly smart glob of jello, that there are sort of specialized areas that have particular sorts of intellectual functions mapped to it. And it's not a free-for-all, right? So damage to one part of the brain can't cause any possible sort of behavioral deficit. There is a lawful mapping between damage at one particular location and some particular set of mental operations that might be impaired. Additionally, you guys have probably heard things like this: oh, we only use 10% of our brain, or I'm creative because I'm right-brained. Now, after having passed through your 1-hour crash course in neuroscience, I know you'll never have to, you'll know to refute those claims when they come up again. We use all of our brain all of the time for different sorts of mental operations, and there really is nothing more to right-left brain organization than language and spatial representation, okay? Okay, so now that we have had our tour of the brain both on a macroscopic large scale and then on a microscopic scale about how individual nerve cells connect to one another and communicate, now we're finally ready to take the brain and stick it under the microscope and start to look at it with some of these imaging techniques. So in the second part of the talk, what I'd like to do is run through some of the different sorts of imaging methods that exist, what they can measure, and what sort of inferences or claims we can make about them. So that's what we're going to cover. And the first division to know about in different sorts of imaging measures that are available is this electrical versus metabolic distinction, right? So now you'll recall that nerve cells communicate to one another first with a fast electrical pulse that travels down the axon, and then a slow chemical communication as the neurotransmitter diffuses, travels from one neuron to the next, right? So there's two sorts of imaging methods, and broadly speaking, or two types of imaging methods, broadly speaking, they are measuring either that electrical activity or that more slow metabolic activity. So electrical imaging has two primary types. There's what's referred to as EEG, or electroencephalography, or MEG, which is magnetoencephalography. And these are techniques that make measurements on the surface of the scalp, right? So either with the little electrodes or with little devices that can detect changes in magnetic field structure. So these are arrayed on the surface of the scalp, and these are then sensitive to the movement of electrical charge down sets of axons through the brain. In general, these sorts of techniques have very good resolution in time, right? So they can measure events very rapidly and with great precision about when they occurred in time, but they're a little fuzzy about where things are happening in space, okay? Because you're measuring a signal from the surface of the scalp, it's hard to know precisely where in the brain that signal came from, but you can say very precisely when it happened. That's in contrast to what we can call the metabolic imaging types. So these are imaging methods which are measuring the changes in kind of metabolic activity that are associated with nerve cells, right? The consumption of oxygen and glucose generally. And within this category falls PET, or positron emission tomography, SPECT, which is single photon emission computed tomography, and functional MRI, which is the one we're going to come back to and discuss the most in the end. So again, these reflect the metabolic activity of neurons. Generally speaking, these measures have relatively poor resolution in time, but great resolution in space. And the reason you might think that they have poor resolution in time is because they're kind of measuring sort of the coal being shoveled into the furnace, right? Not the electrical activity itself that is the communication of the cells, but more kind of the churning through fuel and oxygen that the nerve cells engage in as they engage in these computations. Again, this is a broad overview, and there's an awful lot of messy details that lie just very close beneath the surface. So just to take one imaging method, for example. So positron emission tomography, which I will say nothing more about today, is a technique which can measure many different sorts of metabolic states and many different aspects of neural function. So positron emission tomography can measure the amount of oxygen that's being consumed or the amount of glucose that's being consumed. And with clever design of these sorts of experiments, you can actually measure the neurotransmitters themselves, the concentration of the neurotransmitters, where they're going, how they're being picked up. So there's an awful lot of more subtle aspects of neural metabolic function that can be measured with these techniques beyond the sort of the broad just level of activity that I've been describing so far. Okay, so finally, here's where the rubber hits the road. Let's get right down to what is the preeminent neuroimaging method that exists for modern studies in neuroscience. So bold functional MRI, the nitty-gritty. And what I'd like to do is tell you a little bit about how we go from changes in neural activity to a brain image that we can measure, okay? So the first thing you should know is that, of course, MRI scanners have been around for a long time and used in clinical practice, right? So MRI scanners are used to obtain images of the brain to diagnose structural abnormalities, to see if, for example, somebody has a tumor, if there's markers of multiple sclerosis. And those brain images now are shown in 3 different views. So the brain is a 3-dimensional structure. You can only ever show 2 dimensions of it if you make a picture of it. So you have to just pick which particular direction you would like to view it from, and those different views have different names. What's shown up there is the sagittal or side view, the coronal, and by the way, name for Sagittarius, the archer, always pictured in side view, the coronal or crown view, which is the face-forward view, and then finally the axial or horizontal view, which is the one down at the bottom, okay? So I'll be showing you primarily images in the axial view. So an MRI scanner can be used to measure lots of different types of things. So depending upon the way that the MRI scanner operates, you can measure different types of pictures, obtain different sorts of pictures, and those can be sensitive to all sorts of things. There are ways of setting up the scanner so that you're very good at detecting pockets of blood in the brain, or to measure the flow of blood through arteries, or to identify different sorts of fatty tissue, or different sorts of combinations or concentrations of water. And so all these different operations or scanner settings are devised to help diagnose particular kinds of medical conditions. But the particular application that we're interested in is for measuring neural activity, right? So for measuring not just the structure of the brain, but the function, the moment-to-moment change in the metabolic state of neurons. And for that to work, we need one other thing to be true that thankfully is, and that is that there is some relationship between the neural activity in a set of in an area of the brain and the amount of blood flow that takes place in that area, okay? So a very fortuitous aspect of the organization of the human brain is that it has very good what's called neurovascular coupling. So changes in neural activity, changes in brain activity at one particular location give rise to a change in blood flow at that very same location, right? So all the little blood vessels at that one site get a little bit bigger to let a little bit more blood flow in. And you can think of some very perfectly reasonable explanations for why Mother Nature has this system for us, right? So if nerve cells are increasing their metabolic activity, they need a bit more of a supply of fuel, and they might have a bit more of sort of waste products that need to be flushed away. So increasing blood flow locally is sort of an efficient organizational system for the brain, but also a real godsend for neuroimagers who'd like to study it. And because then we can use the MRI scanner in a manner which is sensitive to the amount of blood flow that's present at a given location. And the way that works is because of another fortuitous property of nature, and that is that the property of blood differs depending upon whether there's more or less oxygen present there. Okay, so here's how it works. In blood, you have molecules called hemoglobin. Hemoglobin is the molecule in your blood which is carrying oxygen. It picks up oxygen in the lungs and delivers it to the tissues. Hemoglobin has inside of it an iron atom, okay? And that iron atom is actually what the oxygen binds onto, okay? Iron, you'll remember, has magnetic properties, right? So if you have like a bar of iron and you take a magnet, clong, it'll stick right to it, right? It turns out that the magnetic properties of iron are slightly different, microscopically different, depending upon whether oxygen is or is not stuck onto that iron atom, okay? So basically rust, which is iron with oxygen on it, rust has slightly different magnetic properties from fresh, clean iron, okay? An MRI scanner is a machine which has a very long coil of superconducting wire which is cooled with liquid helium and generates an enormous magnetic field, okay? So when the college student volunteer comes along, we put them on the scanner and slide them on in, and what we're sliding them into is an unearthly, powerful magnetic field, right? Which I hasten to add has never been shown to have any harmful effects whatsoever. So a very strong magnetic field, and that magnetic field then can be used to measure that property of iron as to whether it's more rusted or not rusted, right? Whether it is carrying oxygen or it isn't carrying oxygen. So what that all adds up to then is our neuroimaging technique, all right? Through a chain of just happy coincidences of physiology and very carefully designed physics, we can create a chain of events which lead from a change in neuronal activity to a brain picture that reflects or measures that neural activity, right? And just to walk you through it, increases in neural activity change the local blood flow, right, as the brain kind of brings in some fresh blood and oxygen. So that change in local blood flow alters the amount of oxygen that's bound onto the hemoglobin, which then alters the magnetic properties of that hemoglobin, which we can then measure with the scanner, okay? So this little causal link chain is what brings us from changes in neural activity to something we can measure with the scanner. And at the end of the day, what we get out is a functional brain image, right? A picture that looks like this, where each point in the picture reflects the relative level of neural activity that was taking place there at that point in time, all right? Now, one thing you'll notice and that we're going to come back to is this picture is a lot fuzzier than those structural brain pictures I was showing before, right? So we saw a set of brain pictures before, the ones that are used for if you're looking to see if there's a tumor in there or if you're looking to see if somebody has multiple sclerosis. Those brain pictures are all very sharp and clear. These brain pictures are a little bit fuzzier, right? In practice, these pictures are made up of tiny little measurements, each about 3 millimeters on a side, okay? So each point of this picture is composed of what's called a voxel, right? So if you go to your TV screen and you get in real close, you can see the individual points that make up the picture, right? And those points are called pixels. As I get older and older, I can't see the pixels anymore. But if you get in really, really close, you can see the pixels. In the same way, these functional brain images are what are made of what are called voxels. And the reason they're called voxels is because they have a volume to them, right? Because we're making a three-dimensional picture. So each little pixel, each point of measurement has a length, a width, and a height. So that's 3 dimensions. And those dimensions are about 3 millimeters on a side. So just to give you a sense of what 3 millimeters looks like, there's a finger with a little 3 millimeter square. I know not everybody here uses the metric system. So that's what 3 millimeters looks like. And inside that 3 millimeters is about 630,000 neurons, right? So by the time you do the math of the number of neurons in the brain, and each little measurement that you make is about 3 millimeters on the side, inside that measurement is about 630,000 neurons, which is, you know, a lot of neurons, but still not too bad, right? We're sampling neural activity at the aggregate level of what, you know, just under 1 million neurons are doing. Okay, and we obtain these pictures over time, right? So you get one picture of the brain with this kind of resolution every 3 millimeters, and you get them about every 2 to 3 seconds, right? So somebody sits in the scanner and they're looking at pictures or they're performing some kind of task, and the scanner is rumbling away and taking one picture of the brain every 2 to 3 seconds over time. So what I'd like to talk to you about now is how you would go from that string of pictures that you've obtained from somebody sitting in the scanner to one of these processed brain images, a brain image that shows some activity and about which you can make some claim about behavior. And it's time finally to refer back to that original case we started off with Brian Dugan, right? So as you'll recall, the claim was that this guy, Brian Dugan, you could show by performing functional MRI that he is a psychopath, right? So what I'd like to do is dig into that claim a little bit and show you kind of what the work is behind the scenes that leads ultimately to that claim that somebody might stand in court and make, okay? So what were the individual measurements made, what were the statistics performed, and what were the analyses done to finally lead to that claim. So we're going to start actually with a different kind of claim, and that's the claim that psychopaths as a group show reduced responses to emotional stimuli within the amygdala, okay? So that's the claim that backstops the one about Brian Dugan, right? So first we're going to start with that claim, and then we'll come back to Brian Dugan. So the amygdala is a structure in the brain that's involved in emotional processing. So it's been known for many years that if you look at pictures of faces which are angry or sad, that there's a response within the amygdala, which is your sort of emotionally valenced response to seeing those stimuli. And the claim is that psychopaths as a group have a different kind of response, a reduced brain cell response within this emotional part of the brain, the amygdala, to seeing these emotional stimuli, okay? So again, here's what we're going to do. We're going to take a subject and we're going to put them in the scanner, and we're going to obtain these measurements over the time, right? So all these voxels, 3 by 3 by 3 millimeters, 630,000 neurons per voxel, and we're going to start to get some measurements over time, all right? So here's a plot of the data that might result from one of these sorts of studies on one person from one point in the brain, okay? So on your left is one slice of the brain, and you can see all the individual voxels indicated in a grid, and that one black dot is just one voxel that we might want to study or look into. And then shown in that plot to the right is that fMRI signal, the neuroimaging signal over time, as somebody sat in the scanner and was shown a series of faces one after the other, okay? So for example, I might get two faces. I might have a picture of a face which is showing the emotional expression of fear, and a picture of a face which has a bit more of a neutral expression. By the way, just between you and me, the neutral face looks kind of angry. But at least supposedly the neutral face is supposed to be, that's supposed to be a neutral face. So you have somebody in the scanner, you put up a picture of the face, a fearful face, a face with emotional content, and you do that for about 30 seconds, and then you have the subject take a look at another set of faces, which are neutral faces, which do not have emotional content, and then back to the fearful ones, and then back to the neutral, and then fearful and neutral, and fearful and neutral, and you switch back and forth between these conditions over the course of the experiment, right, about 30 seconds for each one. And what you might observe for that one point in the brain is that every time the fearful faces were on the screen, the neuroimaging signal went up, and then every time the neutral face was on the screen, the neuroimaging signal went back down, okay? So you might want to try and quantify what that effect was at that one point in the brain, right? So you could average over all the times the fearful faces were on the screen and over all the times the neutral faces were on the screen. You take the average of those two things and you get like that difference between the fearful and the neutral faces for that one point in the brain, right? And you could measure that by any one number of things. You could say it's so many units of neuroimaging difference, or you could do a statistical test and have a statistical value associated with that. And at that point in the brain, you could actually write it down. You could say, well, you know, the effect here was a significance at a level of p less than 0.02. That's what often scientists use to report their results. They say, how unlikely was it that this result occurred by chance? And they apply a probability, right? And it might be, oh, it was only 1 in 100 times if I did this experiment would I have been fooled by noise into thinking that there would have been an effect there. So that's a p of 0.01, all right? So you might write that number down. Well, but you know, you can't actually write down a single little number on the picture like that. That would be tough to see. So what is done instead is you color code it, right? You say, okay, well, what I'll do is I'll put down a colored dot at that point. If it was a small effect, I'll make it kind of red. And if it was a really big effect, I'll make it yellow, right? So you have a color scale for each point. And then you go on and do that same kind of analysis at every single point in the brain. You measure how big of an effect, how big of a difference was there between the fearful or emotional faces and the neutral faces. Boof. So you fill up the whole brain with different colored dots, where each dot is an indication of how big that effect was, how big that difference was between the two groups, okay? Here's then what we get into the realm of what I call neuroaesthetics, which is how we go from that initial analysis to the final resulting figure that might appear in the Newsweek article or in a scientific publication, okay? So we start off with these brain images, which are kind of fuzzy, right? For which we've calculated some statistical effect at each point in the brain. But, you know, it's really hard. You can't see the brain structure underneath all these colored dots. And moreover, most of these colored dots are just noise, right? Because every dot I'm going to compute some kind of measure for, but only some of the dots are actually statistically reliable. I only want the, you know, results where only 1 in 100 times would I have actually been, you know, fooled by noise. I don't want to see anything where it's likely I was fooled by noise. So I'm going to apply a threshold. I'll get rid of all the small effects. So I apply a threshold now. That's much better. So now I can see where the big blobs of activity are that were really the big effects of seeing the fearful faces versus the neutral faces. You know, but the problem is there's still some like little tiny dots around, and what I'm looking for are kind of big smooth effects. So I'm going to apply some digital smoothing. And let me say actually that each of the steps that I'm going to describe along the way, there are very valid statistical reasons for each of the steps I'm going to describe. So thresholding, the math is very well worked out for what kind of threshold you pick and how you do it. Digital smoothing has been shown to enhance your ability to detect some of these effects, so it's generally done. So in any event, so after you apply your digital smoothing, there you go, that's a nice, clean, clear blob. All those little noisy, tiny little voxels are gone. But you know, the problem is I can't really see the activity on that fuzzy brain picture. Luckily, I have a nice, crisp, structural brain picture I acquired at the same time. So I'm going to take that brain activity, right, those colored dots, and I'll display it on top of my nice, crisp, structural brain image instead of my fuzzy, functional brain image. That's much better, right? Now I can see where that activity is in the structure of the brain, and that looks like the insula. Okay, fantastic. But the final problem, of course, is that that's just one slice through the brain, one little horizontal slice here. And of course, the brain has a whole volume. It goes from top to bottom. And I'd like to be able to see all of those slices of the brain. So, you know, I go over to my friends in the computer graphics department. I say, hey, you know, could you show me all these slices at once, maybe like in a 3D view with like a little cutaway so I can see, oh, that's good, right? So now I've got the whole 3D structure of the brain, and I can see the blob located in situ at that location. Okay. Now, again, I hasten to add, every single one of these steps is reasonable and well justified and part of good scientific practice. But I just would like you to be aware of sort of the difference in appearance from the original data and then the final thing that's presented, okay? That what in the end you're seeing in these images is a color-coded representation of a statistical effect, not the brain actually in some sense lighting up, right? There isn't actually some light that happens in there. What's being expressed is a color-coded statistical result, all right? We have yet another problem, of course. This is just the result of just studying just one subject. Suppose we take 10 people and we run them through this experiment. We show them fearful faces and neutral faces. We might end up with a result like this, you know, subjects 1 through 10. Everybody has a blob kind of roughly in the right spot, though. Maybe some folks didn't really get the blob, but everybody looks pretty good. Is there a way maybe we could combine that data across the 10 subjects and show what's in common across them? Okay, well, methods exist to do that. Everybody's brain is slightly different shaped, but we can come up with digital warping routines that gets your brain to kind of squeeze like putty to be shaped like my brain, and we get all the brains lined up, average them together, there we go. That's the result, okay? So let's return to that claim. Psychopaths show reduced responses to emotional stimuli within the amygdala. So what's actually underlying that claim is that a group of people were put into a scanner, some people who were normal controls, some people who had undergone behavioral testing to be identified as psychopaths. And then once those folks were in the scanner, they saw a series of pictures, fearful faces, neutral faces. And then the brain activity, after all their brains were kind of warped into one common space, was measured at this one site, the amygdala. And what was found was that the level of neural activity was greater in the normal control population compared to the set of psychopaths, okay? And an important thing on that plot that I'm showing there is not just what the average was across the group, but also the variability, which are shown with those error bars, right? Because some people had big effects and some people had small effects. But on average, I'd say that if these were your data, that claim would be pretty well supported, right? Yeah, it's definitely the case. There is a difference in the average response between the psychopaths and the normal controls in the mean level of activity within the amygdala. But of course now, and as was alluded to earlier in Owen's opening remarks, we face the very difficult challenge of making this claim, which is that this person is a psychopath because he has reduced responses to emotional stimuli, right? Because what would have to be true is that he would have to have a response which was not just within the normal, which was not just lower than the normal average, but outside of the normal range, right? So that you could be quite certain that he wasn't just a normal person with just a normal variability and a slightly low response. And of course, these aren't problems which are new or unique to neuroimaging, right? So far from it, these kinds of problems have been around for hundreds of years. If people have tried to take group-based research studies and then apply it to individuals. But I'd just like to illustrate them here to point out that neuroimaging, for all of the fantastic technology that's involved, doesn't get around this really fundamental problem of trying to take a group result and apply it back to individuals. In fact, actually a deeper issue, and one that Professor Morse will be touching upon, I think, at some point during this conference, is if psychopathy is defined by behavior, how is the neuroimaging data even relevant, right? And we have to think carefully about what the particular sorts of questions are that neuroimaging provides additional leverage beyond what could have been obtained just by having had that behavioral measure to begin with, right? If you had a behavioral measure that said that Brian Dugan was a psychopath, how does the neuroimaging data tell you something you didn't know, right? And I'm not suggesting that it is not possible for neuroimaging to contribute additional or new information. It's just we have to look carefully and think about how those questions are structured for that to be accomplished. All right. In the final couple of minutes, I just want to point out a couple of ways, generally speaking, that these sorts of inferences or claims are made about neuroimaging data. how we go from having a brain image with some kind of result to a claim like this guy is a psychopath, or for example, this guy is lying. And of course, that last one is a rather pertinent example. There are already companies out there, you can find them on the internet, where you can go and undergo testing with an MRI scanner. They'll charge you for the service and then produce some results, which will give you some indication as to whether or not you were truthful or not by their measures. And attempts, of course, have been made to introduce these results as evidence in cases, in legal cases, but so far I understand have not made it past a Daubert challenge. So I would like to then illustrate just two directions of neuroimaging inference, right? There's a huge world of neuroimaging studies, many about which you'll hear about today, things that you've come into contact before from reading in the lay press, different sorts of mental operations that have been studied and examined. And I just wanted to tell you that all of those different studies boil down to two basic modes or two basic kinds of questions that are being asked about the brain. And each of those sorts of studies has a core assumption, a key assumption that makes the claim possible. So when you encounter one of these studies, you can go right to that key assumption, right to that key claim, and figure out if the study and the claim is well supported or not. So here you go. The two sorts of studies are what are referred to as forward inference studies and reverse inference studies. So a forward inference study might be of the type that we have identified the brain area for pain, right? So that we've identified a little chunk of the brain that this is where the perception of pain takes place, okay? And so that's a forward inference study. And the way that such a study might be done, or the way that the claim looks is you say, well, when someone is experiencing the mental state of pain, then this brain area will show increased activity, right? That's the claim. That's the forward inference study. And the way that that study is actually done, as we've talked about a little bit before and illustrated, is that you isolate a behavior by subtracting two conditions, right? Before we had fearful and neutral faces, and we're looking at the difference between them. So in this kind of study, you might take, again, college student volunteers. It turns out they'll do just about anything for a Starbucks gift card. You put them in the scanner, and then for 30 seconds, you sort of jab them with a needle, and for 30 seconds, you brush them with a feather, and for 30 seconds, you jab them with a needle, and you alternate back and forth between these two conditions, and you see which brain areas activate, okay? And so that is how one of these forward inference studies would be done. And the conclusion then, of course, is that the difference between being jabbed by a needle by the experimenter and being brushed by a feather is this experience of pain, and that's what's driving the activity. Well, you know, the way that studies like this could go wrong is if there are other sorts of behaviors that go along with, you know, the manipulation that you were doing that could also be driving this activation, right? Maybe it's not pain per se, but the anticipation, like, oh, here comes the needle, right? I mean, maybe that's what's driving the activation. Or perhaps anger, like a sense of rage that they were fooled into being in the study, and so every time the needle comes out, now they're getting angry. And that's what's driving the activity. So making those forward inference studies depends upon being able to claim that you have isolated the particular behavior of interest, right? That you really have been able to find some manipulation of experimental states which isolates the pure essence of this particular behavior, in this case pain, okay? That's a forward inference study. Now let's look at a reverse inference study. So a reverse inference study might be of this kind. Does hearing a bad joke induce physical pain, right? And this actually, there's studies have been published along this line, right? So I tell you a real groaner, some lousy pun, do you actually feel a physical sense of pain the same as if you get jabbed by a needle, right? So the way that that study looks is that I tell you the bad joke, and I look to see if there's activity in this area of the brain that I've previously identified as being the pain area of the brain, right? So if I see that neural activity rises in that little chunk of brain when I tell you the bad pun, then I will conclude that you are in the mental state of being in pain. And this is akin to the sort of inference that was being made about that one individual, Brian Dugan, right? We looked in a particular area of the brain, we saw a degree of activation, and we said, there is less emotional response. He is not having an emotional response to fearful pain, to fearful faces, therefore psychopath, right? So in the same kind of inference here, there is increased activity when I tell you that bad joke about the two priests, the rabbi and the nun, and so there's actually a physical sense of pain. Now, this kind of reverse inference study, of course, suffers from its own kind of potential problem, and that is, what if some other kind of behavioral state can activate this region, right? What if it's not just pain that activates this region, but other things like confusion or fear or unpleasant memories or anything else, right? So your ability to conclude that the subject is in a particular behavioral state relies entirely on how well you've proven that that one piece of brain is for that one mental function, and that when you see activity at that location, you're guaranteed that the subject's in a particular behavioral or emotional state, okay? The final example, and this is what I'll end on, is the FMRI as a lie detector. And this is a sort of inferential approach that uses both forward and reverse inference in a combined study. And generally speaking, these are studies which use a technique known as multi-voxel pattern analysis, which is by far the most important development in this field in 20 years. It is the major source of most studies that are being done now, and I think going to be the major driving feature for neuroimaging for the next decade. So I'm going to tell you just very briefly an example of a multi-voxel pattern analysis study. In fact, a study was just published in the New England Journal of Medicine, which showed that you really can deduce quite well from the pattern of brain activity if somebody is experiencing physical pain or not. This is work by the lab of Tor Wager. This has enormous implications in medicine and perhaps in law as well, that if you can have an objective measure of the subjective sense of pain from a scanner, and they have cracked that nut using this multi-voxel pattern approach. But let me give you an example of this as it applies to lie detection. So here's the goal. You want to tell if somebody's lying, right? And the goal is to have the brain, to identify the pattern of brain activity which is associated with lies, okay? Some pattern of activity not at a single point in the brain, but across the entire brain, some pattern of rising or falling neural activity that suggests that the subject is in the state of lying. And the way that such a study might be done is that you have somebody in the scanner and you have them look at cards, and they can either tell the truth about the card they're looking at, or they can tell a lie, right? And you want the scanner to be able to detect when they're telling a lie. And so the way that this kind of study is done in 2 stages, a forward inference study followed by a reverse inference study. So in a forward inference study, you put the subject in the scanner and you ask them to either tell the truth or tell the lie. You say, okay, well look, for the next half an hour, I'd like you to pick cards and lie about them, okay? And then in the half hour after that, I'd like you to pick cards and tell the truth about them, okay? And you then use the computer to look at the pattern of brain activity associated with the lying state and the truth-telling state, and learn the difference between those 2 patterns of brain activity, okay? So that's step 1. So you have them tell the truth about some, and for some you have them lie, and you actually give them the instruction to do this. Step 2 is the reverse inference. You now put them in the scanner and you say, okay, I'm going to put cards up on the screen. You pick. You can lie or tell the truth, but later on you have to tell me which one you did. And I'm going to see if I can use this pattern of brain activity to deduce whether or not you were lying or telling the truth. So you look at the pattern of brain activity. When the subject makes a response, you say, well, that looks an awful lot like the pattern that I saw earlier associated with telling the truth, or that looks an awful lot like the pattern associated when the subject was telling lies about the cards. And so based upon that pattern match, you then say it's either a lie or it's telling the truth, right? So this works. This works for a remarkable number of mental operations that you can use this kind of MVPA, pattern analysis, to deduce mental states about the subject. The real key question, the weakness, the thing you need to ask about these studies is how well they generalize outside of the particular contexts of, in this case, looking at playing cards and being told it's okay to tell a lie or it's okay to tell the truth about the particular cards you're seeing, right? Inferentially, it's a long way away from figuring out if you ever, shot a man in Reno just to watch him die, right? I mean, the difference between being given permission to lie about a card and then all the complicated circumstances that goes along with any real-world lie is a vast gulf. And that is really where the weakness of these studies come in, or the potential problem that needs to be addressed, okay? So just to kind of wrap all that up, what I would say about neuroimaging is that the science of the neuroimaging is astounding, but it really doesn't obviate the need for a logical claim. And I hope I've given you some sense of the sorts of structure that needs to be present to be able to make some claim about these patterns of brain activity that you see in publications all the time. Thanks, and I'm happy to take some questions.